Today I am going to talk about three-dimensional imaging of mouse lever dynamics using polarization-sensitive optical coherence tomography, and I am Pradipta from Computational Optics Group at the University of Tsukuba. Liver is the central metabolic organ of the body. High metabolic activity in liver is supported by an efficient microvasculature and is responsible for providing oxygen and nutrients to different organs. Physiological and structural changes in liver microvasculature are associated with several liver diseases that can trigger the progression from healthy to inflammatory to fibrosis and finally hepatocellular carcinoma. So what are the requirements for liver imaging? First, we need to have a technique that can measure tissue microstructural property. For example, birefringence imaging is sensitive to fibrotic structure, whereas attenuation coefficient is sensitive to tissue density. Second, we need to have a method that can retrieve functional properties such as activity of liver tissue and vasculature. So the method should be able to measure tissue dynamics. Next, we should visualize and monitor changes the activity around microvasculature, and for this we require non-invasive and level-free measurement. Finally, we need few millimeter depth resolve investigation, and this requires three-dimensional measurement. To achieve these requirements, let me first show you the current standard techniques. Taking the liver histology is the gold standard method. However, it is invasive and only a certain cross-section of vascular architecture can be observed. Computed tomography can reveal three-dimensional microvasculature. Unfortunately, it is unsuitable because it provides poor resolution. Fluorescence microscopy can observe tissue dynamics due to injection of dye, however makes it difficult to access the longitudinal changes. And also, it is a two-dimensional imaging modality. OCT-based methods such as OCT angiography is non-invasive and can reveal three-dimensional microvasculature and has been widely used in ophthalmology. This method is only suitable for blood flow imaging and cannot be utilized for tissue dynamics. So our strategy in this research is to use an imaging modality which can visualize and quantify the dynamics around microvasculature of a tissue. Recently, several groups, including ours, has demonstrated and validated tissue dynamics imaging based on OCT that can reveal intercellular motion such as tissue dynamics, and this method is non-invasive, level-free, and can provide three-dimensional tissue dynamics. The details of the method I am going to explain in latter part of my talk. So our purpose in this research is to visualize and quantitate liver microvasculature dynamics using OCT microscopy based dynamics imaging method. Let me explain the method and experimental setup used in this proposed study. To achieve tissue dynamic sensitivity, we captured multiple B scans at the same location of the tissue, and then by computing the variance of time sequence dB scale OCT intensity signal at each pixels, we can visualize tissue dynamics, and we call it as log intensity variance LIV. In the pseudo-color composite image, high dynamics region is appeared as green while low dynamics is appeared as red. However, the LIV imaging is still two-dimensional. And according to our previous study by my colleague given in this reference, we found that the number of frames required to visualize tissue dynamics is not that much. And 16 frames around 6 seconds is enough to visualize tissue dynamics contrast. However, for three-dimensional dynamics imaging, if we capture 16 frames over this time at one location and scan multiple locations, the total acquisition time will be large enough. To overcome this issue, we designed a new three-dimensional scanning protocol. To explain about this, let me first show you the inverse chart. Total 128 locations in the sample is divided into 4 groups. For each group, the raster scan is performed and the system captures 16 frames at one location, and the time separation between the first and last frame is 6.55 seconds. The scan is performed for group 2 to group 3 to group 4 in a similar manner as of group 1. The total acquisition time for a volume is 26.2 seconds, and the total number of frames or B scans used in the study is 2048. In addition to tissue dynamics, we are also interested to tissue microstructural measurement, and for this we require birefringence and attenuation coefficient property of the tissue. To measure these two properties, let's think about the Jones matrix first, which is a purely a mathematical quantity but possesses almost all fundamental optical properties of the tissue. We can derive OCT intensity by applying averaging to the absolute square intensity of the four Jones matrix entries. Then we can derive attenuation coefficient, which represents local scattering property of the tissue from OCT intensity, by applying the Vermeer method. Using eigenvalue analysis, we can retrieve local birefringence, which is sensitive to collagen. Now, your question might be how we can measure the Jones matrix of a sample. The answer is in this optical scheme. The system to retrieve Jones matrix is polarization-sensitive OCT microscopy system. The central wavelength of the light source is 1.3 micron and the lateral resolution is 19 micron, whereas the axial resolution is 14 micron in tissue. The A-scan speed of the system is 50,000 A lines per second. The Jones matrix OCT system is identical to the standard OCT except these two extensions, which are polarization delay unit and polarization diversity detector. Owing to these two extensions, we can acquire 4 OCT images by a single scan. Let me now talk about the study and measurement protocols. To validate microvasculature dynamics, two different studies have been performed. One is healthy mouse liver and another is carbon tetrachloride treated fibrotic liver imaging. Let me first discuss about our first study. For this study, wild-type mouse has been used. Mice were first sacrificed to extract the mouse liver, and then the liver was kept in a culture medium to keep the tissue alive. Then after 1 hour, OCT measurement started at room temperature 24 degrees centigrade, and we measure after every 6 hours until 4 days continuously. Dynamics imaging of healthy mouse liver just after the sacrification is shown here. The lumen was appeared as hyposcattering region in cross-sectional OCT image, whereas the cross-sectional LIV image shows high temporal fluctuation of OCT intensity just beneath the lumen. The in-face image at this depth location shows the vessel structure in OCT intensity, whereas the in-face image just beneath the vessel shows high LIV appearance, and it also appears as vessel-like structure. Cutaway volumetric visualization of LIV shows high dynamics appearance just beneath the vessel at deep regions of mouse liver tissue. To interpret this, let me first show you the acini of liver. This region is the periportal area of liver and is the center of acinus, where oxygen content is highest. This zone has the highest metabolic activity due to respiratory enzymes. Therefore, what we are observing in LIV image just beneath the vessel might be due to this high metabolic region. Although whole periportal region is known to be active, but we don't observe high activity above the vessel, and this remains as an open issue. To validate the activity around the vessel wall region, longitudinal measurement was performed. Time course cross-sectional and in-face image of OCT and LIV at different time points are shown here. At 24-hour time point, high LIV at the vessel region faded and becomes low. We can observe in day 2, 36-hour time point, the vessel region starts to show high LIV again. At very late time points such as day 4, 84-hour time points, high LIV at the vessel region starts to degrade and become low. We can observe the same phenomena in LIV in-face images similar to the cross-sectional images. High LIV appearance again after 36 hours in ex vivo liver is little hard to interpret, as there is no related study found in the literature. To investigate this, we require further study, and it remains as the next step. Please note that the result of the high LIV appearance again after 24 hours is repeatable, as we obtain similar result for another mouse liver sample. Let me now show you our second study on fibrotic liver. For this mouse model, acute liver injury was induced by intraperitoneal injection of CCl4 for continuous 2 days. After that, mouse were sacrificed to extract the mouse liver, and then after 1 hour OCT measurement started, and we measure after every 4 hours until 32 hours continuously. The results of fibrotic liver at 0-hour time point is shown here. There are some isolated high dynamic spots appeared in cross-sectional LIV image that cannot be observed in OCT image. At this depth location, The in-face image of LIV also shows isolated high dynamic spots, and we cannot observe clear vascular structure in the in-face image, which was observed in the case of healthy mouse liver. To further investigate the appearance of these high dynamic spots, LIV volume rendering image was generated by applying intensity thresholding procedure. We can now observe the vessel-like structure that corresponds to the high dynamic spots in LIV images. It is noted here that the vessel, which cannot be observed in OCT image just after the sagrification like healthy liver, LIV image gives high contrast to identify the vessel surrounding tissue. Time-lapse imaging was performed and the results at several time points are shown here. The cross-sectional LIV image show that the high dynamics became dim along time and completely diminishes at 32-hour time point. Similar appearance can be also observed in LIV in-face images. For more quantitative analysis, mean values of OCT intensity, attenuation coefficient, LIV and birefringence are plotted as a function of time. The region of interest is shown by the box in the images. First, the mean LIV is high in healthy liver than fibrotic liver. Mean LIV result reveals three phases of long time course tissue alteration around the healthy liver microvasculature, whereas for fibrotic liver, LIV gradually decreases with time. Although OCT and attenuation coefficient shows some changes at certain time points, it doesn't show any clear tendency to those of LIV. Next, high birefringence was observed in fibrotic liver due to triggering of CCL4, and it was known that the extracellular matrix such as collagen 4 accumulates due to CCL4 injection. In addition, the mean birefringence shows some long time course changes. The increased nature of birefringence can be occurred due to increase of collagen density, and it might be due to liver tissue shrinking. While working for the study, we found that the different optical contrasts reveal several tissue properties. Therefore, we may require a synthesized tissue contrast that may represent all the tissue properties at once. So what we need is tissue contrast synthesis. Our approach for tissue contrast synthesis is by applying principal component analysis to the dataset that contains all the original optical contrasts such as attenuation coefficient, LIV and birefringence. Each pixel of a contrast is unfolded to form a one-dimensional vector, and then these three one-dimensional vectors are arranged in a matrix where each column represents each contrast. Then we take this matrix and do PCA on it. In PCA method, the principal components are essentially the eigenvalues of the covariance matrix. By using eigenanalysis, the eigenvalues and eigenvectors can be determined. The first principal component has the highest eigenvalue, and this corresponding eigenvector will have the highest variance. By folded back the first principal component to the original position of the pixel, we can determine the synthesized tissue contrast which has the highest variance between all the input features. For demonstration, the method as described previously is applied to the volumetric data of healthy mouse liver. As expected, the first principal component shows higher variance, almost 50%, while the variance is lower and is least in second and third principal components respectively. The first two principal components represent almost 80% of the input data. Contribution of each feature to the principal component is represented by their feature scores. The feature scores of LIV and birefringence has much higher value than attenuation coefficient to the first principal component, while it is lower in second principal component. The cross-sectional and in-face images of the three principal components of normal mouse liver at 0-hour time point are shown here. To enable direct visualization of the distribution of three PCs within the tissue, each PC was assigned to a color channel within an RGB image to generate a final composite image representation. Some recognizable feature can be seen here. The surface of the tissue and the vessel appeared as magenta. The tissue just beneath the vessel is appeared as yellow, which is the contribution of first and third PCs. The higher depth region appeared as red, while the depth region below the surface appeared as green. Let me now conclude my talk. We demonstrate tissue activity around liver microvasculature using OCT microscopy based dynamics imaging method, and it was validated by longitudinal visualization and quantification. As a conclusion, the volumetric dynamics imaging based on OCT could be feasible to visualize and monitor changes the activity around microvasculature or mouse liver and can complement the current standard modalities. Here I would like to thank my COG colleagues, co-authors and our funding agency. Thank you for your attention.